Hello students, welcome to the third lecture of the chapter Molecular Basis of Inheritance. I am Mouchandra and let's begin. So quickly recap what we have studied in the previous class. So we saw Griffith's transforming principle. Okay, so Griffith did his experiments on two strains of pneumonia causing bacteria, that is Streptococcus pneumoniae, the R strain and S strain. And we know that he saw that there was something called transforming principle that got transferred from the heat-killed S-strain bacteria to live R-strain bacteria, which caused the transformation of R-strain bacteria into S-strain, because of which the mice who were injected with a mixture of heat-killed S-strain bacteria and live R-strain bacteria, those mice died. Okay, so his experiment is also called a transformation. Okay, so his experiment is still done today to genetically transform various kind of bacterial and mammalian cells. Next, we saw Avery, MacLeod, and McCarthy's experiment. Okay, so they actually try to analyze what is the biochemical nature of this transforming principle that Griffith proposed. Okay, so they found that DNA is the genetic material which actually got transferred from the heat-killed S-strain bacteria to live R-strain bacteria, because of which the R-strain bacteria has transformed into a virulent type and caused the disease pneumonia. All right. So that, but scientists at that time, biologists at that time, they were not convinced with every MacLeod and McCarthy's conclusion. Okay. So people were still debating. People were still debating at that time whether a genetic material is a DNA or is a protein. All right. So then came Hershey and Chase, who did the experiment that is called transduction experiment using bacteriophage viruses. All right. And their experiment conclusively proved without any doubt that DNA is in fact genetic material, okay, which contains all genetic information and gets passed on to the next generation of the cells. All right. So what did they do? They used bacteriophages who are known to infect E. coli bacteria, and they saw that the DNA material from the bacteriophage was getting was entering the bacterial cell, not the protein of the bacteriophage. So from that, this they concluded that DNA is a genetic material, and it was conclusively proven. And scientists after that, biologists after that, didn't argue, and it was accepted universally that DNA is genetic material. All right. But people have seen in later studies that RNA is also genetic material in very primitive type of organisms like viruses. Okay. So many viruses like coronavirus, they have RNA as a genetic material. So we then looked into what are the criteria or the ideal properties that a genetic material should have. All right. Also, we compared DNA and RNA individually, as both, if as we know that both individually can act as genetic material in different organisms, but all multicellular higher organisms, okay, they have DNA as a genetic material. RNA is seen only in few viruses. Okay, so let's quickly recall what are the criteria or the properties for genetic material. We saw that genetic material should be able to replicate itself or generate its own replicates or replica. Then it should be stable enough, both chemically as well as structurally. So in the second point, we saw that RNA is a very reactive molecule in comparison to the DNA, and we know the reasons, right? So the third point for the criteria of genetic material was we saw that DNA or the genetic material should be able to, you know, should be able to get expressed in form of Mendelian characters, right? Genes, so that it can pass on from one generation to the next generation. And lastly, we saw that genetic material should allow the introduction of some changes, but the introduction of these changes should happen slowly. These changes are what we call mutations, which we'll talk about in more detail in this chapter. Okay. So, and we know that mutations, they are the seed for the evolution, which we'll again cover in the next chapter. Okay. So these were the few things that we saw in previous class. So in today's class, we will focus on the replication or the DNA replication, or how does DNA replicates. So if you remember the central dogma of molecular biology, we know that DNA, the information from DNA gets copied into RNA, right? And information from RNA gets copied into proteins. So the copying of information from DNA to RNA is called transcription. Formation of protein from RNA is called translation, and DNA can create its own copies, or can create its own replica, and this process is called replication. So first we will cover this process, replication, and then in the next classes we will individually discuss transcription and translation. So after the discovery of the DNA structure by Watson and Crick, okay, people now started discussing how the DNA is replicating, or how DNA is making its own copies. So three models for DNA replications have been proposed. Okay. So what are these three models? First model is called the semi-conservative type. Second model is called conservative type of replication. Another is the dispersive. The semi-conservative, conservative, and dispersive. So these are the three models of data replication that people had proposed at that time. Okay. And we'll now later see in this discussion which of these three models is actually seen in nature. Okay. Or is actually proven to be the correct model for DNA replication. So let's understand each of these replication models individually. So I will read each of these definitions. Okay. So first is the semi-conservative replication. So what happens in this replication is that each strand of the original DNA molecule, it serves as a template for the synthesis of new complementary strand. So as a result, each new DNA molecule contains one original strand and one newly synthesized strand. Okay. So there is the parent DNA molecule. Okay. And we know in a DNA molecule, there are two strands. So each of these parental DNA strands will act as a template for the synthesis of the new complementary strand. So as a result, we will get now two DNA molecules, and each DNA molecule will have one copy of parental strand and one copy of newly synthesized strand. Okay. So it will become clear from this particular image. So here, the left side is showing, sorry, not this one, this. The, for the middle one, the middle image is showing the semi-conservative type of replication. So here, these two gray strands, these are the parental strands, let's call it as P. Okay. And when DNA is replicating, what we are getting as a result, two DNA strands. From one DNA molecule, we are getting two DNA molecule, and each new DNA molecule, we see one parental DNA strand, and another is the newly synthesized complementary strand, the blue one. Okay. In the similarly, there will be one newly synthesized complementary strand, and another is the parental strand. So this model of DNA replication is called semi-conservative. Now, another model has been proposed that is called conservative replication. So here what happens, the two original strands or the parental strands of the DNA molecule, it serves as templates for the synthesis of new strands. Okay. Now, after the new strands has been synthesized, what happens? These two original strands, they come together to form the original DNA molecule, okay? And the newly synthesized strands, they come together to form a new DNA molecule. As a result, the original DNA molecule remains intact. So again, let's go back. So this this image, which I've labeled as 2, is showing the conservative model for DNA replication. So these two are the parental DNA strands, okay, or the original DNA molecule. So after DNA replication, we'll get two DNA molecule, correct? A copy, another copy will be generated. But here what happens is that the uh, original DNA strands after replication, they have come together, okay? So this is the original one. They have come together and made the identical strand which was there at this stage. Whereas the newly synthesized complementary strands, which are labeled as NN or the blue color, now they have come together to form that new DNA molecule. So this is the conservative model for DNA replication. So in conservative model, the original DNA molecule, it remains intact. But in the semi-conservative, what we are saying that one copy of the original strand, it combines with one copy of the newly synthesized DNA strand. Okay, so this is semi-conservative, this is conservative. Now there is a third model which is called dispersive replication. So here what happens, the original DNA molecule, it gets broken into fragments, all right? And then each fragment, it serves as template for the synthesis of new complementary strand, all right? So the resulting DNA molecule will contain both original as well as newly synthesized fragments. So here what is happening, this is the original, this is third image. So this is the original DNA molecule or the parental DNA molecule. Now it gets fragmented, okay? Fragmented. So here you can see the two new DNA molecules. The gray ones are the original fragmented DNA molecule. And each fragmented DNA molecule is serving as a template. So if this is the original parental one, and if this parental fragment, DNA fragment, will act as a template to which a complementary fragment, okay, will be synthesized. And this will be like this, okay, and similarly, a checker box kind of a pattern one can see, and this is called dispersive model of DNA replication. So we saw all the three models: semi-conservative, conservative, and dispersive replication models. All right. Now we have to know which model is actually the accepted one. Okay. Universally accepted model for DNA replication, or how does DNA replicates in the cells. So we need a experimental proof, okay, which shows which of these models, the semi-conservative, conservative, and dispersive model, which of these models is actually seen in nature. So one experiment was done by two people, Matthew Melson and Franklin Stoll, okay, in 1958. And they experimentally proved that DNA actually replicates in semi-conservative manner, okay, which means the new DNA molecules will have one copy of the original strand and one copy of the newly synthesized complementary strand. So how did they prove experimentally? We will discuss their experiments. So they used Escherichia coli, which is the bacteria, you know, or E. coli, and they studied the replication mode in this particular bacterium. And then later on, perform the same experiment in plant cells as well as in human cells. So first they took E. coli and grew them in a nutrient media which contained ammonium chloride, right? So what's so special about this ammonium chloride? This ammonium chloride contained N15 isotope of nitrogen. Now this N15 isotope is not radioactive. In fact, it is a heavy isotope. Okay, uh, heavy in the sense it is more denser than the normally found nitrogen molecule. Okay, the normal one is N14. It is not radioactive. Please note, no radioactivity. So this N15 nitrogen molecule is not at all radioactive. So this E. coli, few colonies of E. coli were grown in a nutrient media which contained ammonium chloride, and this ammonium chloride, the nitrogen is of N15 type. Okay. So what will happen? Okay. If so, these E. coli, they will in their cells, the replication process will happen. Correct. Uh, New DNA strands are synthesized. So while it is happening, N15 molecule, okay, and N15 will get incorporated, okay, it will get incorporated in their DNA molecules. So we know in a nucleotide there are nitrogenous base, sugar, and phosphate group. A nitrogenous base consists mainly of nitrogen groups. Now in all these E. coli cells, within these E. coli cells, N15 will be there in their nitrogenous bases, because of which the DNA molecule will be heavy. Okay, it will be more denser than the normal one. And not only N15 will get incorporated in the nitrogenous bases, but it will also get incorporated in various nitrogen containing compounds, okay, which are normally produced inside the cell. So if you know, or if you recall Hershey and Chase's experiment, they labeled the nitrogen, okay, radioactive labeling they did for the nitrogen to get labeled proteins. So from that experiment, as well as we know the structure of the amino acid, where amino group is there. So all the proteins in the cell, in the E. coli cell, will also get labeled by this N15 isotope. But we are not worried about that here, okay? We are only concerned with the labeling of the newly synthesized DNA molecules, okay, with this N15 isotope, okay? And so I'm labeling the N15 DNA molecule with red, okay? This is the heavy DNA molecule. It will have a higher density because of the presence of the heavy nitrogen isotope. Now what they did, they then took a few colonies of the E. coli, which they initially grew in N, N15 ammonium chloride containing media. Okay. They took few colonies of E. coli which were grown in the media containing ammonium chloride with N15 isotope, and they were transferred into another nutrient media. Now this another nutrient media contained ammonium chloride but with N14 isotope. That is the normal one. Is normally found in the nature. So in another medium where the E. coli were transferred, it contained ammonium chloride, but the nitrogen is the normal isotope, N14 isotope. So now when in this new media, in this new nutrient media, the E. coli will replicate, right? It will use that media to divide and, make more and more E. coli cells. So when that will happen, the N14 isotope will now get incorporated into the newly synthesized DNA molecules, because of which the newly synthesized DNA molecule will contain N14 isotope instead of N15. Okay, till now it was straightforward. Now what they did, they're now going to analyze the DNA molecules that were formed, okay, in the E. coli cells that were grown in N15 NH4Cl media, and the E. coli cells that were, grown in the media containing ammonium chloride with N14 isotope. So they took, extracted the DNA, okay, from these E. coli cells, and what they know initially, like what was given to them or before starting their work, that E. coli takes only 20 minutes, okay, takes only 20 minutes to divide. So every 20 minutes, a new generation of E. coli cells will be formed. So, so what they did, they took few E. coli cells initially, okay, so at time 0, okay, and extracted their DNA molecules. Then waited for 20 minutes to get generation 1 of the E. coli cells, took those E. coli cells, extracted DNA molecules. So in this way, they studied, they analyzed the DNA molecules that are formed in every generation, okay, that is in every 20 minutes. And what they did, they took those DNA molecule and centrifuged it. Okay, they centrifuged it in a cesium chloride density gradient. So cesium chloride is a solution within which they added the DNA molecules extracted from the E. coli, okay, and then centrifuged it. If you know what is centrifugation, where we apply very high speed rotational force, okay, so because of which gravity pulls down the heavy molecules at the bottom of the tube, whereas the light molecules, they float, okay, at the top. So in this way, they separated the DNA molecules basis of their density, on the base of the density. What is the density of the DNA molecule? Using cesium chloride solution. So after centrifugation, what happened? Each DNA sample or a sample taken from the E. coli cells from every generation, they separated independently, okay, on the cesium chloride gradient, depending on their densities. So whatever was the density of the DNA molecules that they had, okay, they separated and formed bands, individual bands within the solution, okay, and these bands were observed only after they centrifuged. Before, it was not observed. So this is a representation of that, this image, okay. So if this is a tube, say, and this white color is the cesium chloride solution, so the light DNA, which will contain only the normal N14 isotope, it will form a band at the top, okay, because it is light. Then it is not very dense, okay, it has low density. Whereas the DNA molecule containing only N15 type of isotope, which is the heavy isotope, it will form a band at the lowest most section, okay, the bottom section of the tube. Whereas if the DNA molecule contains both N15 as well as N14 isotopes, a mixture of both, then the density will be in between between these two extremes. Okay, it will be between these two extremes. So let's call it as hybrid. Okay, so so from this we can expect 3 types of DNA molecules: light DNA, hybrid DNA, and heavy DNA, and they will get separated based on their densities within the cesium chloride solution after centrifugation. So this is called cesium chloride density gradient. Okay, so here you are able to see that how they are using heavy isotopes, okay, not the radioactive isotopes, in order to distinguish between the DNA molecules only based on the densities. So since we are studying what is the mode of DNA replication, okay, it's really helped them. Okay, how? Let's look at their observation. So this image shows what was the result or what they observed at the end, okay, after the centrifugation. So if this is the heavy isotope. Okay, so initially when E. coli was grown in the media containing N15 isotope, okay, at that time, all the DNA molecule of the E. coli contained only N15 isotope. So the DNA molecule, it was heavy. So because after centrifugation, the band was at the bottom, okay, at the bottom, okay, because it was heavy. It is more dense. Whereas after 20 minutes, first generation of E. coli will be obtained. And here, at this point, they used N14 isotope, correct? They enriched the medium with ammonium chloride containing N14 isotope. So after 20 minutes, when they analyzed the DNA samples from these E. coli cells, they saw that a band was obtained, but it was towards the up, towards this side, okay? Uh, top, top of the, it was above the uh, this red color band, which means it is showing a hybrid DNA, means a mixture of both N14 and N15 is present. Okay, means if we draw it like this, here is the heavy one, which they saw initially, whereas the hybrid one will be above the heavy one. Okay, and then just again analyze the DNA sample after next 20 minutes or 40 minutes, taking this is T0, this is T20, and this is T40, T is equal to 40. So if, and they took DNA samples from the second generation of the E. coli cells and observed bands of light DNA molecules. The light DNA molecules will be here containing only N14 isotopes, okay? As well as they found a band of N14 and N15 as hybrid, okay? So, and the thickness of the band was equal, okay? So this was the result. Now, this result was only possible if the DNA is replicating in a semi-conservative manner, means the newly formed DNA molecule will contain one copy of both the parental DNA molecule and one copy of the newly synthesized DNA molecule. So initially, the original DNA molecules contained only N15 isotope. Okay. Now, after 20 minutes, the newly synthesized DNA molecule contained or will contain N14 isotope. Since the DNA is replicating in semi-conservative manner, the newly formed DNA molecule will contain both original one as well as the new one. Original one will have N15 isotope and the new one will have N14 isotope, which is why they observe a hybrid band, okay? A band which was in between the two extremes, okay? Which is why this hybrid is possible. Whereas after 40 minutes, okay, this for second generation, each of these, say this green strand, okay, is the N14 one, and the red strand here is the N15 one. Now, each of these strands in both this DNA molecule is acting as a template strand, okay, or acting as a parent. It is the parent strand to which a complementary strand will be synthesized. Okay, that is why if you imagine it, this green one here, this green one is acting as a template. And since in the media there is still N14 isotope, so the newly synthesized strand will have the N14 isotope in it. So we'll get a DNA double helix containing only N14. Whereas when the red strand here is acting as a template to which a newly synthesized strand will be present, okay, and it will contain only N14. So we get a hybrid strand. Same will be for the other strand as well, because of which we get equal number of N14 containing DNA strands, an equal number of N15 containing hybrid N15 and N14 containing DNA strands. So can you now imagine what would happen if they studied the DNA samples after 80 minutes? Okay. Will they get equal a proportion of light and the hybrid strands? So answer is no. They won't. They will get an unequal proportion. Why? So we have to draw it. Okay. So this is after 80 minutes. That is third generation of the E. coli. So this is second generation from which this is second generation from which we are getting the third generation E. coli. Now, this first strand, or the first DNA molecule, each of the strands will act as a template, okay, and to which a complementary strand will be synthesized. So because of which we'll get 2 copies of light DNA. So this is 1 copy, this is 2 copy. 2 copies of light DNA will be formed. Now the second strand, second DNA molecule. One strand is containing N15 isotope, another strand is containing N14 isotope. So when the DNA is replicating semi-conservatively, the light strand, okay, the N14 containing strand, to which a light strand will be synthesized. So we'll get a 1 molecule of light DNA, okay, and another molecule will be a hybrid type, okay. Why? Because the red DNA strand, which is containing N15 isotope, okay. So to that, DNA strand will be synthesized, which will contain only N14. So because of which we'll get 1 hybrid DNA strand, another will be light. Same argument will be for this DNA molecule as well, okay. So that will give rise to 1 hybrid DNA molecule and 1 light DNA molecule, and you can then similarly expect that this 4th DNA molecule will give rise to 2 light DNA molecules, correct? So when you see the proportion of the light DNA molecule containing only N14 isotope is higher than the hybrid DNA molecule, which contains both N14 and N15 isotope. So one can expect something like this of a density gradient, cesium chloride density gradient, when you do the centrifugation, after doing centrifugation. So this experiment conclusively proves that DNA replicates in a semi-conservative manner. Okay. What would now? My next question is, why did they use cesium chloride solution? So cesium chloride solution actually allows the formation of a density of increasing density, means it allows the formation of a gradient of increasing density. The molecule or the DNA molecule having the lowest density will be at the top, whereas the molecule having the highest density will be at the bottom. So in this way, you can get a gradient, okay, from low to high density, okay, within the cesium chloride solution. So that will then allow separation of molecules containing or having different densities, okay. So the molecule having high density will be at the bottom. The molecule will be which has low density will be at the top, okay. So in this way, the solution is such that allows this kind of a gradient formation. That is why they chose cesium chloride to analyze the DNA molecules containing 2 different isotopes of nitrogen. Okay. Next, so as I told you that Messelson and Stahl's experiment conclusively proved that DNA replicates in semi-conservative manner. Okay, means hybrid DNA molecules were formed, or the DNA molecules having density in the intermediate range of the 2 extremes was observed in their density gradient. Now, what result would you expect if DNA replication was happening in a conservative manner or in dispersive manner? Recall what is conservative. In conservative manner, the original DNA molecule will remain intact. Okay. Whereas in dispersive manner, we will get only DNA molecules containing both fragments of original as well as newly synthesized. So the conservative DNA replication would gives a result, a hypothetical result of something like this. Okay. So initially, when E. coli was grown in media containing N15 isotope, all the DNA molecule will be heavy, right? Will contain only N15 isotope. Now, after 20 minutes in media containing N14 isotope, what would happen? We won't see any hybrid DNA molecules. Means we won't see any band corresponding to DNA molecules containing both N14 and N15. Rather, we will see one band for DNA molecule containing N14 isotope and one band for the DNA molecule containing N15 isotope. Same result we will see after 40 minutes as well. Only difference would be that the thickness of the band will increase. Okay. We won't see any hybrid DNA molecule. Okay. Whereas if the replication were say dispersive in nature, okay, means the original DNA strands get fragmented and that each fragment is acting as a template for synthesis of the new fragment of DNA molecule, new fragment of DNA. So in that case, initially, yes, we will get all the DNA molecules containing only N15 isotope, that is, they will be heavy in nature. Now, after 20 minutes, we will see hybrids, right? Hybrids that contains both N15 and N14. After 40 minutes, also we will see only hybrids. Only difference is the thickness of the band will increase after 40 minutes. The same will be for 80 minutes as well. Means in dispersive, the original DNA molecule is lost. Okay, as well as we don't see a band for only the newly synthesized DNA molecule. Means we don't see a green color band. Only we are seeing in subsequent generation bands for the hybrid DNA molecule. So since none of these cases arise, like the result that Messelson and Stoll saw, it is not what one would expect for the conservative and dispersive. So again, through this argument, I can also say that Messelson and Stoll showed the data replication to be semi-conservative. Whatever result uh, Messelson and Stoll saw here is only possible if the DNA was replicated in semi-conservative manner, where both hybrid as well as newly synthesized DNA molecule will see. Okay. Here, the original DNA molecule is lost. Okay, after 3, 4, 5 generation, we can see only hybrids or newly synthesized DNA molecules, green या फिर brown bands. So that is another way of arguing why DNA replication is semi-conservative. Okay. Now let's talk about the mechanism. How does DNA replicates, or what things are necessary for DNA to replicate inside the cell? We will understand it through few points. First point is that DNA replicates in S phase of cell cycle. All right. Uh, S phase, if you remember the cell cycle that you studied in class 11, there was G1 phase, S phase, G2 phase, M phase. So in this S phase, if you remember what happens, and from the original copy of the DNA, a new copy of the DNA is formed, right? So if there is n number of chromosomes, it becomes 2n. The number doubles, or the content of the DNA, it becomes doubled. So how it is doubling? It is doubling because DNA replication was happening or happens in the S phase. So DNA replication always happens during S phase, and it happens prior to prior to cell division. Okay, so it is a preparation phase when cell is making its arrangements for itself to get divided into two daughter cells. So then the DNA replication happens. For DNA to replicate, it requires these following things. First, it needs the DNA, obviously. That is, it needs um, template to which a complementary strand can be synthesized. So the original DNA molecule, each of the DNA strands, there are two DNA strands in a DNA molecule. So each of these DNA strands will act as a template to which complementary DNA strands will be synthesized. Then it also needs monomers or deoxynucleoside triphosphates. Okay, so DNA is a polymer, and to this formation of this polymer, you need the monomers. So these are the monomers which will get joined together by phosphodiester bonds, and you get a chain, polynucleotide chain. Then what you need an enzyme, okay, that will catalyze this polymerization reaction, make this reaction go fast, means it will happen at a faster rate, and this enzyme is called DNA dependent DNA polymerase. All right, so you need these three things for and before DNA replication begins. So for replication to begin, first, what should happen? The double-stranded DNA helix, it should first unwind. Okay, so it is in helical form, so it should unwind in the opposite direction, so that each of the two DNA strands get exposed. Okay, their nitrogenous base sequence, they get exposed, and this unwinding is quite energy expensive process. Okay, it requires a lot of energy, ATP molecules it needs. So that is why uh, unwinding doesn't happen for the entire DNA molecule at once. So in humans, the DNA is 2 meter long. So if every time, suppose during replication, uh, energy that is energy is used up in just unwinding, okay, the entire 2 meter long DNA molecule, then energy is also needed for the survival of the cell, for other functions of the cell, or it is also needed for the subsequent steps of the replication. Then ATPs won't be available, right, for the next steps. So what actually happens? Only a small portion of the DNA molecule gets unwind at a time. Okay, and it forms what is called a replication fork, which is a Y-shaped, something like this. A Y-shaped structure gets formed, but only in a small portion of the DNA molecule. Okay, so certain enzymes are responsible for this unwinding. These enzymes are called helicase enzymes. Okay, and these enzymes require energy. Okay, energy in the form of ATP to do the unwinding process. Okay, uh, means rotate the DNA helix in the opposite direction, so that the nitrogenous base sequence, okay, inside the DNA molecule get exposed. Okay, now replication fork cannot form randomly. It cannot form randomly at any place. Okay, it cannot form randomly. It has to form at some definite regions within the DNA molecule, and these regions are called origin of replication. So these regions, the origin of replication, these are present at some interval. Okay, and it is present throughout the length of the DNA molecule, and only at this origin of replication the replication fork will be formed, means the helicase enzyme will come, bind to the origin of replication, and then cause unwinding, thereafter forming the fork kind of structure, that is Y-shaped structure. Okay. Now after, so this is say a DNA molecule. Okay. Let's say this is the origin of replication. So the enzymes that catalyzes the unwinding, that is the helicase enzymes, they come here and unwind or the rotate the helix in the opposite direction, thereby exposing these nitrogenous bases. Okay. So here, now in the next step, when the polymerase enzyme will come, it can now easily read these nitrogenous bases, which are marked with red. Okay. And after reading, it can then appropriately add the monomers, okay, the nucleotides, in a complementary manner, right? If there is A, the new strand T will come. If there is G, in the new strand C will come. So in this way, the new strand will be formed, which will be complementary to the original strand. Okay. So this Y-shaped structure is called replication fork, and as this formation of replication fork and the catalyzed formation of the new DNA strand, it happens simultaneously. Okay. So that it doesn't take more time. It happens very fast, the DNA replication process. Hardly in E. coli, it hardly takes means 18 minutes. Okay. So in 18 minutes, the entire DNA molecule has to be replicated. A new copy of DNA molecule has to be formed. So to do that, it happens simultaneously. And the direction of formation of this replication fork, it continues in 5' to 3' direction. Okay. If this is 5', this is 3'. Okay. It happens. Okay. Because of which and also the DNA polymerase, it moves in this direction only, the 5' to 3'. And as the DNA polymerase is moving, the replication fork is also forming, and the new DNA strand is getting formed in this 5' to 3' direction. Now, DNA is a polymer, as I already told you. It is a polymer of deoxynucleotides, right? And the enzyme, DNA polymerase, it is a DNA dependent polymerase enzymes. Enzyme means it requires DNA as a template for the synthesis of the new DNA strand. It cannot use an RNA molecule. Okay, it only needs a DNA molecule as a template for the synthesis of the new DNA strand. And this polymerase enzyme catalyzes the polymerization reaction. Okay, or the polymerization or the addition of new deoxynucleotide strands and forms the phosphodiester bond. So that is what this enzyme do. Now, this speciality of this enzyme is that it is a highly efficient enzyme. Means it works very, very fast. So let's look at how fast it works. So in E. coli, the length of the entire DNA of an E. coli is around 4.6 million base pairs. Okay. And from Michelson and Stoll experiment, we know that E. coli divides within 20 minutes. So in every 20 minutes, a new generation of E. coli is formed. Whereas within this 20 minutes, 18 minutes is only for the replication process, replication of the DNA. Okay. 18 minutes is used to replicate the entire length of the DNA. So if you see, it takes, so this enzyme, DNA polymerase enzyme, or DNA dependent DNA polymerase enzyme, it works at such a fast rate that it can add 2000 base pairs, okay, per second to the new to the synthesizing strand of the polynucleotide chain, which is the average rate of the polymerization that the DNA polymerase shows inside E. coli. So that is how fast this enzyme should work. Okay, in order to replicate the entire length, which is a million base pair long. So you can imagine in humans, which contains billion, right? 6.6 into 19 billion base pairs. So here, in human cells, there are also polymerase enzymes. And now these polymerase enzymes also have to work very fast. So it human cell, say from skin tissue, it can take 3 to 4 days, okay, to replicate and all. So accordingly, there will be a particular rate of polymerization associated to the DNA polymerase enzyme. So the point is, these enzymes have to work very fast. They have to be very efficient in nature. Now, it's fine that it is fast, but it also has to make sure the accuracy means it cannot add a new nucleotide at a place where it was not supposed to be added. Okay, it cannot make any mistakes, because if it makes any mistake, then mutation will happen. Okay, the newly synthesized strand will have the sequence which is not complementary to the original sequence, and when this new strand gets propagated into the next cell, it will then manifest itself in form of a disease. Maybe if it can manifest itself in form of a new phenotype. If a plant should supposed to have green leaves, say, because of this mutation it starts to have yellow leaves. So what I'm trying to say, that the polymerase enzyme should have to make sure that there is no mutation, means it should accurately copy the DNA sequence in the template into the newly synthesized DNA strand. Okay, and the monomers, as I told, are the deoxynucleoside triphosphates. Now these triphosphates have two roles. First, obviously, to act as monomers, which is deoxynucleoside monophosphate or the nucleotide. These nucleotides get added at the 3' OH end, okay, by the DNA polymerase enzyme, okay, and in this way, chain of polynucleotide chain of polynucleotide means many nucleotides get formed, right? And the bond, a phosphodiester bond is formed between the 3' OH group and the 5' phosphate group, which you remember from the first class. So that bond is made by the DNA polymerase, and at the 3' end, these nucleotides gets added. So the formation of this phosphodiester bond, it actually requires a lot of energy. So from where this energy will come? So these monomers, deoxynucleoside triphosphates, they have another role. What is that another role? Their role is to provide energy. So monophosphate form of this compound gets added, right? So two other phosphate groups, they are additional. What happens to them? They actually get the other two phosphate bonds get broken. So the breaking of those two additional phosphate bonds releases a lot of energy, and that energy is sufficient for catalyzing the formation of the phosphodiester bond within the newly synthesizing nucleotide chain. So deoxynucleoside triphosphate, it actually provides energy in the same way ATP does. Okay, because of the breaking of the phosphate bonds. So here phosphate bonds are getting broken. That releases energy, and that helps this polymerase enzyme, DNA polymerase enzyme, to add new nucleotides to the growing polynucleotide chain. So if suppose this region is ori, okay, and this is the original the parental DNA molecule, okay, and as I told you that replication always happens from the 5' end to 3' end direction. Means the DNA polymerase can synthesize the new strand only from the 5' end to 3' end direction. So if this is the ori, here the replication fork, the Y-shaped structure will get formed. Okay, and because of which the bases on the inner side of the two parental strands get exposed, which can now be read by the DNA polymerase enzyme. Okay, and by after reading the sequence, it will appropriately and accurately add the new nucleotides and form phosphodiester bonds. Correct. Now, what happens here? This strand, which is running from 3' end to 5' end, when this strand is the template, the synthesis can begin. Synthesis of the new strand can begin from this end only, right? So the DNA polymerase now can add the nucleotides from 5' end to 3' end direction. So when this is happening, this strand is called the leading strand, and this way of synthesizing the DNA molecule is called continuous. Okay, the DNA strand is getting synthesized in a continuous manner at the template strand, which is of 3' to 5' end. But the problem happens, or the situation becomes more difficult for the template, which is running from 5' end to 3' end. So here, the synthesis cannot begin at this point, okay? Because we have to maintain that anti-parallel manner, okay, of the DNA molecule. The DNA strands are anti-parallel in nature. So we have to maintain that rule. That rule has to be followed. So, which is why the synthesis of the new DNA strands cannot happen at this 5' end. It has to happen at this 3' end. But replication fork is here. So at this particular uh, template strand, which is running from 5' end to 3' end, the synthesis of the new strand happens in a discontinuous manner. Okay, why discontinuous? Because the DNA strand gets synthesized fragment by fragment. Okay, so one fragment gets synthesized, then another fragment gets synthesized. Okay, 1, 2, 3. So in this way, one fragment of DNA molecule is synthesized at a time. The DNA polymerase has to, do double job, means extra work for synthesizing each of these fragments on their own. Okay, and it cannot join these two fragments together. For that, another enzyme is needed that is called DNA ligase enzyme. So DNA ligase enzymes then comes later after the fragments are formed, and it forms phosphodiester bonds between these fragments, and thus a continuous strand gets made. So these fragments are named as Okazaki fragments after the scientist who discovered this type of replication. So his name was Okazaki. He was Japanese. So he named these fragments as Okazaki fragments. And this strand, which is gets synthesized with respect to the template strand that is running from 5' to 3', this strand is called a lagging strand. Okay, so this is how the DNA replicates. Okay, where each of the original strands, they are acting as template to which a newly synthesized strand gets made. Okay, one strand is made continuously, another strand gets made discontinuously. So this is semi-conservative in nature. Okay, because two DNA molecules are formed. Each of the DNA molecule has the parental or the original strand and the newly synthesized strand. The semi-conservative, and synthesis on the template strand is running from 3' to 5'. Here The synthesis is happening in a continuous manner, and this strand is called leading strand, whereas the synthesis of the DNA on the template running from 5 prime to 3 prime, here the synthesis is happening in discontinuous manner, and this strand is called lagging strand. And in lagging strand, Okazaki fragments get made and they get joined, or phosphodiester bonds are made by another enzyme, that is DNA ligase. So extra work is needed for the formation of the lagging strand, whereas synthesis of the leading strand, it happens smoothly. So this is how DNA replication happens in cell, and you know what all things are needed. We need the polymerase enzyme, DNA polymerase enzyme, the monomer, the nucleotides that get added, right? Deoxynucleoside triphosphate. And what else we need? Template. Obviously, we need template. Okay. Without template, uh, a new DNA strand cannot be made. So we need these three things. Now, DNA replication is actually a highly coordinated or highly regulated process. Okay. Because if suppose DNA has replicated, means another copy of DNA is now present in the cell. Now this cell is now have gone to the mitosis phase of the cell cycle, where it is fated to divide and make two daughter cells. If suppose that doesn't happen, okay, then what will happen is that one cell will be there where there will be, say, one copy of DNA and another copy of DNA. So a cell containing more than one copy of the whole genome gets formed. And this type of abnormality is called polyploidy, which we saw in the last chapter, right? So this kind of thing usually happen in various kind of plant cells. So here, if the plant cell, say, has 2n chromosomal number, because of polyploidy, where the cell um, failed to divide after the DNA replication, it gains another copy of the genome. So now the total chromosome of the cell is now 3n. So similarly, it can be 4n, etc. So you can now understand how, and if, say, it happens in the humans, then it will be very, very fatal. That fetus or that embryo would not even survive if something like that happens. It is highly fatal. So to make the things happen normal manner, there are various machinery involved which regulates the entire DNA replication process. Okay, that makes sure that there is no mistakes, make sure that the rate of the replication, it should happen consistently, that rate should not change. And yes, something like this polyploidy shouldn't happen. So if you guys, any of you guys decide to study or opt for a field like biotechnology, there you will read courses where your professors will teach in more detail the DNA replication process. But for now, this is enough for your exams and your board exams and your NEET. That is all for the DNA replication process. In the next class, we'll talk about the transcription process. That is how the information from the DNA gets copied into RNA. Okay. Thank you.